Well, good morning. Thank you for coming. Um, yeah, it's funny when you give a title to somebody for a conference, and then you think, oh, I've got a long time to give that and prepare it. Then all of a sudden you haven't, and then you think, oh, I'll do it when I get to the conference. And then all of a sudden you find you don't have time there either, and then all of a sudden you're up till 2 o'clock in the morning trying to complete the presentation for today. So there's going to be areas that I have missed. I'll try and mention those. I'll try and give you a flavour of some of the work that we're doing at Glasgow on real-time detection of bacteria for improving the decontamination efficiency within the food industry. So, first of all, I'll describe what I mean by real-time. It seems to be a term that's used quite loosely by lots of different people. And then an introduction to sort of the problem, some of which relates to the talk that I gave on Monday, which was a rather impromptu one, but that would give you some idea of some of the work we're doing at Glasgow. And then look at real-time detection and decontamination in more detail. Some detection methods. This is where there's a few holes. There's other methods out there that I'm not describing. Then I'll talk about some of the work we're doing at Glasgow, which is laser biodynamic speckle analysis. So that's using lasers to try and look at bacterial contamination. Bioluminescent systems, where we did some real-time work looking at E. coli lux, which is not an ideal model, but it allowed us to at least investigate some ideas. And then sort of the standard microscopy and image processing systems, where, again, you may have some ability to look at real-time detection. And then I'll describe sort of some real-time detection and decontamination systems that we've looked at, or at least in principle we've looked at. So what is real-time? That's the wiki definition. People seem to be using wiki. I don't know whether that's a good idea for some things, but in the absence of any optical illusions, I will declare at the beginning there was no optical illusions in this presentation. I was desperate to put one in, and I completely forgot last night. So if you look at the screen through blurred eyes, you probably won't see anything. So real-time then is basically how we respond to an event. So it very much depends upon what that event is and gathering information on that event over that period of time. So if you're looking at bacteria, obviously their growth is quite small. So if you're just interested in growth on a surface, for example, as some people are, looking at the shape of colony formation, for example, then it's quite easy to collect some information on that over the period of time of growth. However, if you've got a food processing line and you're looking at trying to detect some contaminants on that line, it's quite likely the product is moving at a fast speed. So the problem of actually real-time detection, in allowing you to then do something about that, is extremely difficult within the food processing area. So one common definition for real-time detection is probably going at a video camera. Video camera? Do they exist now? A CCD camera, a camera on your phone, that type of rep rate that they go at. So it means acquiring an image and then processing something about that image before you capture the next image. So it's quite a fast rate that we're looking at to collect data. So as part of an introduction here, some of that data was from the other day. I've added a bit more. So food poisoning in the UK, significant cost. Interesting, the UK figures are only 1% of that, which I don't understand. Well, a bit more than 1%. And there's about 5,000 deaths in the USA, 500 in the UK. So 10%, that's probably about right. And there's a lot of food waste. So if we can have areas to assess in real time bacterial contamination within this food processing environment, and then be able to make a decision as what to do with that data, then we may be able to address some of these figures that we have there. So consumers obviously need safe food. We don't want to eat poor quality food. and ideally a good shelf life. So the question is, can we detect contaminants in real time and inactivate pathogens in real time as well, and within that food processing context? So within a lab, I think we probably can. Can we take that to a food processing plant? That's another question. So what we need in order to achieve this is real-time detection of the pathogens, and also then a decontamination system that we can control in real time, and to be able to vary that depending upon what type of pathogens we have. So the advantages of that are that we can minimally process the food, or maybe the production line, whatever particular point we're looking at within that processing plant. And I'll talk about some other advantages in a little while. So for real-time detection, if we're looking at 25 hertz as the frame rate, 25 images per second, assuming that we can collect that data very quickly, then roughly we have 1/25 of a second, or 0.04, or 40 milliseconds to process that data and make a decision before the next frame is grabbed. So we have a finite time before we move on to the next image. Now that frame rate might not be high enough. You might need a higher frame rate, in which case your processing time is reduced. So depending upon the surface area of detection, do we have a laser pointer? Oh, we do. Okay, so if we got some magical camera that is able to detect microorganisms, the world is a nice and safer place. So we can irradiate, let's say we've got some food processing surface. We're interested in decontaminating that, so we've got some decontamination system here, some magical camera here that's picking up any contaminants on that surface. So we collect the data, we look at processing that data, and then we can use some output signal to control the decontamination system to ensure that we're eliminating any contaminants on that surface. So what do we have out there that can maybe address this? There's a lot of detection systems, and there's a lot of detection systems that are not in real time. So you might take your sample. So general standard protocols are typically 24 hours where you're swabbing and then growing up. The advantages of those systems are that they're going to be accurate. You can look at bacterial screening so you know what the contaminants are. But the disadvantages, your production line may be contaminated, and you don't know that until later. So it's important to have some more rapid techniques. So if you have a real-time detection system, if we can get them working, then you're probably going to have some improved knowledge, but you're probably going to lack specificity over what the contaminants are. So the longer you wait, probably the more information you have about what those contaminants actually are. And the advantages of this process is that you can have an efficient decontamination system, so you're only providing the energy that you need in order to kill the organisms that are there. So this is going to save energy, and also you're going to increase your sterility assurance. You're going to make sure that you get rid of any contaminants on that line. So let's differentiate real-time detection from the definition, how I've defined it here, with non-real-time systems. So here, as I mentioned, you can identify the pathogens accurately, and then you can take appropriate action. But there's that delay, that time delay between having that information and then responding to that information. So you might have to shut the plant down, get a bit of good clean. That's obviously going to be expensive. You're not getting product out the door, so there's going to be an issue there with your bottom line. And the problem may well have occurred before you even know about it. So if we can identify pathogens in real time, we're going to have a shorter shutdown time, or we might even be able to eliminate that entirely by taking the appropriate action as that information becomes available. And the problem, that's as close as I could get to an optical illusion, which I don't think it probably is by the definition. But we have a saying in this country, I'm sure lots of you heard of it, you may well have it in your country, about looking for a needle in a haystack. Now, I Googled that last night, and this image came up, so apologies to whoever did this. I'm sure there's some copyright infringement here, but I thought it was a good image to include. So this is really what our problem is. We're looking for a needle in that haystack, where the haystack is the food processing environment, the needle is the microorganism. Now, if the microorganism was this size, well, it'd be in a different world entirely, wouldn't it? It'd probably be quite scary, I guess. But obviously, they're not this size, and the problem is a lot worse than this. So we are looking for something probably smaller than a needle in a bigger haystack. So onto detection methods. These are sort of ones that are sort of fairly standard. So Myers and Misra, lots of different variants of that. But basically, you take a sample, you transfer this to liquid or agar plate. So you've got some swabbing. And even now, there's probably discussion over transfer efficiency from your swab to everything else and all these other issues. You're looking at overnight incubation. You can check for growth. You can do some screening. But obviously, this is the problem here, overnight incubation. And one of the reasons why I started to look at real-time detection was based upon the work I discussed on Monday, where we're looking at laser decontamination, for example. It was very annoying for me, coming from a physics background, to have to wait 24 hours to get a result. Normally, you tweak something and you can see the result instantly. So we started to look at how we could get information more quickly on that. And there were some areas that we looked at. With my background in lasers, it seemed sort of suitable to go down the optical route. So, and there's more rapid methods than this. A lot of you will know more about this than I do, but the real-time PCR method, real-time PCR, now for me, that isn't really real-time. You're still taking a sample, you're probably adding something to that, you're looking at some fluorescence within that sample, and then you're getting some data back from that. So I'd be interested in what the users, which I guess a lot of you are. How many of you use PCR? Yeah. What sort of time scale is that normally? Half hour, 1 hour. It's like, too long. So I'm interested in something that's quicker than that. Now, whether that can be speeded up, whether you can automate some of those processes and cut that down to minutes, I guess for some applications you probably can. But again, it comes down to the specificity that you want over the infection, if we call it that, or the contamination, and bacterial numbers. So I'm not going to spend a long time on that. There's a lot of good literature out there that describes this a lot better than I can. Real-time PCR detection, for example. So that paper is looking at fluorescent-based methods for nucleic acid analysis in real time. This real time is the phrase that I have the problem with. That's the mechanism. There's another area called FISH, fishing for bacteria in food. This is quite an interesting paper. I found this and I thought, this would be interesting. This will solve all of our problems. But again, this is looking at fluorescence in situ, hybridization, hence the acronym FISH, and it represents a rapid and highly specific technique for whole cell detection. Fantastic. But the problem is after an overnight enrichment. So although it offers this real-time approach, there's still this overnight protocol that you need to use in order to get data from that. But the approach is quite interesting. You can look at sort of placing various biomarkers or things that are going to respond to contaminants on the end of something that fluoresces, for example, and then detect that fluorescence. So there's lots of processes out there that offer the ability for moving towards something that's more real time. But they're very specific. This came up on my Facebook link the other day, as have a lot of images of this conference. I don't know where they're coming from, but it's good to see. So there's lots of photos. If you join Oslam's Facebook, you can see those. So this came up. I don't know how aware you are of this black light feature, but if you have UVA being emitted by an LED, a UV fluorescent lamp that's filtered, for example, and then you look in the optical-ish region, you can see a fluorescence induced in that sample. So the advantage of this is it's large area. So this is very similar to forensic scientists, what they use for detecting protein and other bodily fluids at crime scenes, for example, and you can see splatter on that, so I'm sure you're familiar with those processes. Thank you to this person. That was an awful long citation, but needed that to show you this image of a scorpion, which has this fluorescent property. And this appeared on my Facebook, where you can actually turn your phone into a camera that can detect this. So I haven't tried this. I don't know if it works, but I thought it was interesting. So they just had a couple of different pens, a blue and a mauve one, and they made a couple of filters writing on the back of some sellotape or plastic, coated that stuck that to the lens, and took a picture of a toilet, which isn't very pleasant. So I'd be interested if anyone actually does that and it does work. But you can see here what the problem is. So you're collecting an image, you're collecting a lot of detailed information on contaminants, and you can do something about that. So I'm going to go into talk about some of the areas that we've looked where we could possibly develop real-time detection systems and allow this philosophy to sort of propagate into the food processing industry. So one of the first areas we looked at was biodynamic speckle. So if you have a laser beam, we've got one here. Unfortunately, I can't make that bigger. I should have thought of that earlier. I could have done a trick with a laser beam, but alas, I haven't. So if you look at a laser beam, because that laser beam is coherent, and if it's interacting with a surface that has a roughness associated with it, you're going to get regions of constructive and destructive interference. And if you move your head slightly, if that being stationary, you can see that there's a granularity, there's a graininess associated with that, that image. So here, if that's on a inorganic surface, that image is going to be stationary unless you move your head. You can see the movement. If you're radiating a living tissue or some fruit or vegetable with that, for example, then you can see that speckle pattern change over a period of time. So there's scattering centers within that organic matter that are moving, and that movement introduces constructive and destructive interference with the reflected laser beam. And that movement gives you a shift in the interference pattern in that laser reflected beam. And then if you analyze that, you can understand to some extent what's going on within that sample. So we started to have a look at this application for a number of different areas, including detection of bacteria, but we also used it as a measure of the freshness of various fruit and vegetables as well. And it was actually quite efficient at that. I'm not showing that data today. So I Googled that last night, and one of my papers came up. I thought, oh, this is good, this works. But I didn't really want to look at that because I know that anyway, but I thought I'd tell you about it anyway. So that was a paper some time ago looking at, well, some inactivation techniques, some of the methods I've spoken about, and also detection of bacteria using this speckle technique. So what we have is a low-power helium-neon laser, so that's in the red end of the spectrum, not the green. Oh, sorry, shining this in your eyes, not a good idea. We have a helium-neon laser here and a focusing lens, and we have a cuvette here and a CCD camera. So we can collect over sort of quite a wide area, depending upon what diameter that beam is. So you could put optics in here, for example, to expand the beam. We were looking at maybe focusing it through this material onto the CCD camera. So it's a very simple optical system. And that's obviously an advantage if you're looking at integrating something into a complex production line. So the disadvantage with this technique is the amount, or it's correlating the signals that you get with the information you want to look at. So this is quite an idealized experiment where in this cuvette we placed some microalgae, and we then put into that some hibitane just to look at the variation in the signal that we're getting from the speckle pattern to identify what the decontamination was with the addition of the hibitane. So we added various strengths of hibitane. It was E. coli in suspension, and we had a speckle pattern on here. I could dump it into a computer, look at it on a TV screen, and then do some analysis and output some data from that. So potentially, depending upon what you got here, whatever your decontamination system is, okay, so here it's chemical, but we could look at modifying that to improve the decontamination efficiency. So what do we get? And this is the problem. It's quite difficult to see what you get. So the data is very difficult to analyze. So you're looking at a diffraction pattern over quite a wide area, and that's changing quite rapidly. But fortunately, there's quite a few optical techniques that you can use that allow you to process images relatively quickly. So there's nothing in here that's fluorescing. We're not adding anything to make the sample fluoresce. So it's just taking the straight cuvette with the E. coli in it and looking at the speckle pattern induced by probing it just with a laser beam at 630 nanometers in the red end of the spectrum, a helium neon laser. So we've got the broad level here. So we're looking at something called the temporal contrast, which is basically the difference between the peak pixel value and the lower pixel value. And again, there's various ways you can do this. You can do it over the whole image. You can average up over a certain number of pixels, then average that up over an image. So you can develop an image that would give you the temporal contrast image for the speckle image that you're looking at. So there's complex ways that you can begin to map the distribution of bacteria in real time over that surface area. So it's moving towards something that is potentially interesting and applicable to the food industry. So we then diluted that quite a lot, and probably it's easier if we skip to point E here, where we had sort of the hibitane from the bottle going straight into the cuvette, and you can see that there's a significant reduction in that temporal contrast. And in between those two extremes, you got sort of a few graphs kicking off that identify that you're getting some reduction in activity of those E. coli cells. So that was time, 3 seconds. We're probably collecting data, and you're approaching sort of 25 hertz, if not quicker. So with some of these techniques, with this technique, with a camera, you can obviously collect the data over all of the pixels, over that two-dimensional area, or you could look at a particular line. If you look at a single line from that CCD, it can process the data even more rapidly, but you're losing the spatial information then over that 2D image. So this was another system that we developed. So here we're looking at E. coli lux. So we had a bioluminescent system, and we were looking at the effect of ozone on the E. coli lux. So we chose the E. coli lux just because we could see how they might respond to the treatment of ozone. So we had, this was a bit of a problem because the ozone is obviously quite aggressive, and we had in this system a detector called a photomultiplier tube, which is extremely sensitive to light. So sensitive that with some of them, if you leave them out in daylight, they could take 24 or 48 hours to actually recover. They give you an output signal, and they become saturated. So you have to be very careful with them. And they're fairly expensive. You can get cheaper ones now, but we were looking at trying to avoid sort of too much damage occurring to this. And so what we ended up with was a couple of chambers where we were flooding ozone in equally. And on one side, we did the ozone concentration measurement. So there we just had a UV source and a detector. And then by looking at the amount of UV absorption, you can determine the ozone concentration within that chamber. And then in the other chamber, we were looking at the bioluminescent output from the E. coli lux. So we very simply just put it onto an agar plate. We had a chopper in here, and that's basically just a mechanical chopper, so you're modulating that, the light coming from the sample, and it just allows us to process that data much more accurately with what's called a lock-in amplifier. So by using that, you get a much shorter signal, and it allows you to eliminate a lot of the noise coming from that process. So you get a cleaner signal, and it's more accurate in terms of what you're trying to measure. And here, we just sort of fed those into the oscilloscope, so we could get coincident readings between the bioluminescence emitted by the E. coli lux and also the ozone concentration in this chamber here. So then we could process that on the computer. If we wanted to, we could have had something to feed data back into here, but we were more interested in what was the response of the E. coli lux to the ozone treatment. So we did that, and this is a sort of a typical graph that we had here. So the ozone concentration increased. We've got some arbitrary units here just for convenience. And then it declined. That was a control, and this was a treated sample. So the control didn't receive any ozone treatment. And you can sort of see the variation in the E. coli lux. But here, we can see that with the ozone treatment, there was a massive reduction in the E. coli lux and the radiation being emitted, or the bioluminescence of that sample. The problem then becomes, what does that actually mean? And this was then another problem. So you have E. coli lux, and again, there's probably people in here that know a lot more about that than I do. What does it actually mean? Because if you vary the metabolism of the E. coli, then they're going to change their bioluminescent properties anyway. So although we had some interesting data, and again, this was essentially in real time that we were collecting this data, it's interpreting that data. What does that actually mean? So you probably need to do some other experiments to see if you definitely have got cell death, and to correlate that with the E. coli lux. And someone asked me the other day about recovery. You need to see if the cells are recovering. So there's lots of issues there about these types of processes. But I suppose the E. coli lux or something similar could be used as a biomarker within a food processing environment, where you're passing that through and maybe detecting fluorescence or something else from that process. So I mentioned briefly about the image processing, and this work wasn't quite looking at cell death initially. What we were interested in here was quantifying the morphology dynamics of Euglena gracilis. It's a nice, fairly sort of big-sized organism, so it was useful to look at that under the microscope. But it's also fairly dynamic in terms of its morphology, so it changes shape quite rapidly, or the order of fractions of a second. It can be spherical, it can be elongate, oval, lots of different shapes. So we wrote this paper on some of this work, and it's looking at using a microscope within the system, but then the problem is you might be able to get some high degree of specificity, but you're looking at a much smaller volume. So if you're interested in the large conveyor belt, this is going to be a difficult technique. So you may have to look at sampling and then look at statistics of the data that you get from that and the possible inference on what your whole production line would be, but then you might miss something. So it's problematic. So we did quite a lot of different analysis here. That just gives you an idea of some of the things you can do. So you've got an original image. You can apply what's called filters to that. So everyone with their phones nowadays, all of these filters are generally built into some app that you can get. So you can increase the contrast, you can look at detecting the edge and all these different issues. And then you can do some clever stuff to get into what's called the spatial domain of those images. So you have the normal image that you have, but you can actually invert that image into the inverse space domain, it's called. And you can do that by something called a fast Fourier transform, or lots of other transforms that are out there. And then you can end up with an image like this. And then you can compare that image to maybe some original image. So if you take a reference image, and then you've got another image at a later date, you can begin to see what are the changes from that image to your reference image very quickly. Again, depending upon the size of the image that you have and how much processing you need to do. So you can do a correlation of those images using those optical processing techniques, and it's a whole area where people have done a lot of work. So with these images and different stages of these images, we try to assess what was the best processing for those images. And while this is good, it's quite specific to the system you're looking at. So if you change your system, you might need another type of processing algorithm to actually get the data that you want from that image. So again, it's problematic. Nothing seems to be easy in this domain. So we had a look at trying to quantify the shape of the euglena as it changed. And what was interesting here, this was with one of my PhD students, Anand, who now works at a laser company. And we had a lot of discussion over the order of these, from going from a spherical to an elongate thing. And me and him, we just couldn't agree what the order should be here. So we set up a panel, would you believe, to try and identify what order these should go in. So we had, I think we asked about 10 people, so it wasn't very big. And it wasn't double blind with fancy stuff like you were talking about yesterday, Michael. But I wish I knew at the time, because I'm sure we could have done a much better experiment. It's more like a case of arrange those in order of spherical to elongate. And so even humans are disagreeing on the order of that process. And that made it harder to try and identify what was maybe an optimal algorithm to agree with human perception, which may be the wrong way around. Maybe you need a method of quantifying it independent of human perception, because after yesterday and all the optical illusions we looked at, I don't really trust our vision anymore. So we went from the broadest down to the elongated, and we had, this is not any sense along here, it's just a histogram. So it's just for these different images. But then we contrasted, tried to sort of correlate that with the actual dimensions of the major minor axis. But again, this was a problem. Where do you pick that? So it's an issue. We looked at what's called the cross-correlation. So we were referencing everything to this image, basically. And we were looking at different parts in that analysis here and taking different images and trying to find the best fit for the filter. But what was interesting that came out from this work, just looking at the variation of the pixels over time, gave us a measure of cell death. So the cells are moving, and then depending upon what we have here, this shows the intensity standard deviation. And again, as a function of time. And we were collecting lots of data here. So we were running at real-time speeds, but we're only showing some of it just to show what's happening. So this is as the organism is changing shape. So we can assess the morphology dynamics via this process. Had a problem getting that to agree with human perception. But then when the cells had died, basically through a drying or evaporation process, then there was a significant difference in certainly the intensity standard deviation. So we were looking at the pixel value and looking at how rapidly that pixel value changed over time for this data here. And looking at that data when the cells were dead, the values were significantly different than when the cell was alive. So taking an image, somehow converting that to something else, and then identifying mechanisms and processes that are going on within that image might allow you to develop sort of these real-time detection techniques. Okay, so the decontamination system. Hopefully most of you are here on Monday when I did my impromptu talk. And I'm very quickly going to show you some of those images again, just in case you weren't here. But what I didn't talk about on Monday was the timescale, I think I didn't anyway, the timescale over which this energy is applied. So we spoke about using lasers for looking at inactivation on different substrates. So we've got an ND-YAG laser that's at 1.06 microns. This is about 400 watts, so it's a fairly big laser. And again, we're not applying any photosensitizers, which may enhance that process, so it's direct use of a laser beam to kill microorganisms. I seem to be an advocate of not having to add things to get things to work, unless it's another process to enhance the efficiency of that process. So we looked at nylon, stainless steel, and glass, and the important thing here is the energy density. So that's the joules per centimeter squared that we apply to the substrate. But what isn't in this data is the timescale over which that's applied. So So each pulse, it's a pulse laser, each pulse is the order of milliseconds long. So with the order of milliseconds, you can contaminate an area. That was with a CO2 laser. These were the systems we had. So I spoke about the rate of inactivation as a function of translation velocity for this system and this laser scanning system here. So we have a laser beam coming in. This was just being rotated. It was just a convenient way of getting a very fast laser beam moving over a surface. We had an agar plate here, which we contaminated with collagen. Not contaminated, sorry, we put collagen on it and then contaminated it with Staph aureus. And also we had an agar plates with Staph aureus. And we compared the rate of inactivation just by looking at the area of clearing that we had here after 24 hours of incubation. And the important thing here is the rate at which we're getting this rate of inactivation. So it's centimeters squared per second. So yeah, it's very quick. 100, 150 centimeters squared per second. And we weren't shy on the bacterial loading here either. It's probably sort of 10 to the 6 CFU per milliliter, or 10 to the 8 of that order. So it was significant. And there was no evidence of recovery of the Staph aureus. So very fast, efficient process. If you go too slow, you're supplying too much energy, and you're not at your optimum. If you go too fast, you're not supplying enough energy over that area. So somewhere you're going to have a peak translational velocity, which gives you, an optimal translation velocity for given conditions that gives you the peak rate of inactivation for that process. So I think I showed you this as well. So here we can see that the scanning speed is too slow. Here it's about right, or maybe too high. You don't know. And here we had Serratia marcescens, which is great because you contaminate a sample with that. You can actually see where the clearing is. And that was on some ham. So this was a system that we had. And what would be useful, which at this point I will declare we didn't do, but just to get the idea across, would you have something in here, a detector, some conditioning electronics to look at that, what's going on down here. You might need another laser here, depending upon what you're looking at here. So if you used some of the speckle techniques, for example, you'd need another laser over here, probably, say the helium neon laser. You detect your information, go to some conditioning electronics where you're processing that, and you can feed that back into your processor, and then look at controlling the oscillating mirror here, or maybe the rotary table here. So we had a rotary table and an oscillating mirror above that. So for those of you that remember the old record players, the long players and those things, it was that sort of spiral motion you get. I believe they're coming back in fashion now, aren't they? Okay, so that takes us on to some work that I did with another PhD student of mine, Wiam Jafar, who's finished. And one thing, well, what we were looking at particularly was cleaning of air, decontamination of air using laser systems, which I haven't spoken about at all, but we were looking at modifying the laser to enhance the decontamination efficiency of a laser at cleaning air. And we were looking at Bacillus atrophius, which is like an anthrax simulant. So we'd decontaminate the air and then collect the air samples on an air collector, and then look at the counts that we were collecting. So we were developing laser and plasma systems from some of that work. So with this, we had an Exma laser, so that was at 248 nanometers, so it's well in the UV end of the spectrum. And we were irradiating an agar plate. So again, this is a very simple method, and it's a good way of comparing the efficacy of different processes. So we irradiated an agar plate, incubated that overnight, and looked at the area clearing. So as we've seen, you get this area of clearing. And then what we also did, we looked at focusing any scattered light into a spectrometer through a fiber optic cable. So it's a very simple experiment, very simple. And this is a picture of that image, well, an image of the system, picture of the image. And we've got some focusing lens here. That's the agar plate just mounted. We mounted it vertically for no other reason than it was easier than bending the laser beam down 45 degrees. And this is the detector. Well, the mount for the fiber optic cable, which you can just see coming out here, which then plugs into the spectrometer. So this is what we ended up with. An intensity variation, that's the actual laser beam there. And there's probably three areas of potential interest here where we're getting some information about the spores that were on this agar plate. So the spores, not in the vegetative state spores, so they're going to be probably harder to detect. And there were probably about 3 regions, possibly 4, where we might be able to extract some information to feed into a real-time detection system. So we have an agar plate as a reference. So that was just the agar plate. We needed to make sure that what the fluorescence was on that, or if there was any. There was nothing significant there from, well, it's probably a bit higher than 200, up to sort of the infrared region. But when we looked at the agar plate inoculated with the spores, while we were decontaminating, there was obvious variation in the width of the reflected signal, which could be attributable to the laser beam. And also, there were a couple of peaks here that are probably due to the protein and other parts of the spore. And what I was looking for last night, and unfortunately couldn't find, was a table of the properties of the spore and the fluorescent properties with UV excitation. So basically, we're using the UV laser for killing the spores, but also using that as a probe beam to detect what's happening to the spores on the sample. And I seem to have run out of speed when I was doing the conclusions last night and didn't add any this morning. So in conclusion then, we've looked at a range of potential real-time detection systems. So rapid decontamination systems exist. I just gave you the example of a laser, but you could use any system, microwave, chemical, whatever, high pressure. And the speed of that decontamination process obviously depends on the substrate. The autofluorescence is an interesting approach as a means to measure the degree of contamination. And there's lots of other areas that I've not had time to look at that may offer a higher degree of specificity, including ferrotransform infrared spectroscopy, various other types of spectroscopy, and maybe fluorescence as well. So thank you very much.
So do we have any questions? Dr. Watson, please.
You mentioned that the pixel value varied when you make photos of your experiments. What do you mean? Which is the best value of pixels?
The best value of the pixels?
Yes.
Okay. I've closed my thing down. Why did I do that? I don't know. That was silly. The best value of the pixel, there is really no best value. It depends upon how you're analyzing that data. So it's really the algorithms that you use to process the data which are important. That's where the development work has to be done, really. And that, as I said, it depends upon the system that you've got. And if you change that system, you may well have to change those algorithms. So it is a non-complicated, it is a complicated approach, but offers the potential to offer a significant advance and improvement in what we can come to do. Yeah, well, again, there's lots of different results in there that I showed. Why did I close that down? That was silly. On the graph with the histograms where I had the dead cells, for example, that was looking at the mean of sort of various intensities across that pixel image. So it's not a case of there being a best value. There's a value that describes the state of the organisms that you're looking at. So when we were looking at Euglena, for example, it's a motile organism. It changes its morphology. And as it changes its morphology, then the pixel value varies as well. But what was interesting, that pixel value peaked when the cells were dead. So that's an interesting area that we probably should look at in more detail, really.
Thank you very much. So, very short question, please.
Actually, very short, 2 questions. So, would work only on the surface of the food, not so if the bacteria is already multiplying inside, would that have any effect? And the second question would be, would that affect the nutrient value of the food being decontaminated?
Okay, to take your second one first, I can't see the nutrient effects, the nutrients being affected by the detection process, possibly by the decontamination process, depending upon what decontamination process you use. And to answer your first question very quickly, yes, it tends to be more of a surface effect because we're looking at this problem optically. So it might be good for surfaces. Conveyor belt or something, or food surfaces. But looking within the food, yeah, nightmare. Very difficult problem.